Hello, my name is Angelika Amon. I'm a professor in the biology department at MIT, and I'm also an investigator of the Howard Hughes Medical Institute. In part 1, I introduced aneuploidy and its impact on human health to you. In part 2 of this series, I told you a little bit about the work that we're doing in the lab. And in this part, the third part, I would like to put aneuploidy in the context of human diseases. And again, I would like to, with work from my lab, illustrate to you how we think about the role of aneuploidy in human diseases, especially cancer. And I want to raise some possibilities by which we can use or exploit aneuploidy, perhaps even in developing new therapies for this disease. Like all my other talks, I have basically subdivided this part of the talk into a few subsections. I will first discuss with you the role of aneuploidy in cancer, how we think about it, and how we're going to elucidate its role in these terrible diseases. I will then sort of raise the possibility that perhaps aneuploidy could be developed as a new therapeutic target in the disease. So in the second part of my talk, I try to convince you that aneuploidy is bad news for cells. It causes them stress, it causes imbalance in the genome, and that is associated with a lot of detrimental features, and it basically leads to a stress response. So I hope by now you're actually sitting there and asking yourself, well, if aneuploidy is so bad for cells, if it antagonizes cell division, how come that most solid human tumors are aneuploid? And that's really a question that the field has grappled with for a long period of time. And there's many answers out there, and many people have different ideas about this. Here are my 2 favorite hypotheses, okay? The first one is best summarized by a German proverb that goes, "In the land of the blind, the one-eyed is king." Let me explain to you what that means. Even if you're not very good at something, for example, seeing because you only have one eye, if no one around you can't see at all, i.e. is blind, you will still be top dog. And that's how you can consider effects of aneuploidy. For example, if in an organ of terminally differentiated cells, if no cell can divide at all, then the one cell that acquired an extra copy of MYC because it had an extra copy of the chromosome that the MYC gene is located on, now acquired the ability to proliferate, even though it proliferates badly because this extra copy of MYC comes with all the baggage that's associated with the entire extra chromosome, it will still prevail. So one idea that a lot of researchers, and including us, are very excited about is this idea that under some specific conditions, aneuploidy can be advantageous even though it interferes with proliferation. And there's a number of wonderful examples for this from experimental evolution studies. And I've given you an example before, the fluconazole resistance in Candida albicans that I discussed in part 2. And so you could envision that aneuploidy might be a quick and effective way of providing new traits to quickly adapt to a new environment. For example, adapting to a new niche during metastasis and so forth. So aneuploidy could provide ways to allow you to adapt to a new environment. The other idea that we were intrigued by and that we actually set out to experimentally test, and I'm going to show you a few data about this, is the idea that because aneuploidy causes protein imbalances, that because proteins that are important for, copying and segregating the genome may be imbalanced, and that could cause defects, and it could lead to genomic instability. And so to test this hypothesis, we asked a very simple question: the disomic yeast strains, this series of yeast strains that we developed that carry one particular extra chromosome, do they show increased genome instability? And I'm showing you here a large table, and I don't want you to look at every single one. You can do that later. What I want to do now is I want to just show you that this is the series of disomic yeast strains that we looked at. And then each column represents one assay for genome instability. We looked for increased chromosome missegregation, mutation rates, and various signs of DNA damage. And I'll come back to those in a minute. And what I would like you to take in is that there is a ton of pluses on this table. And what that tells you is that every single disomic yeast strain at least shows one form of genomic instability. Okay? So this idea that whole chromosomal aneuploidy basically induces genome instability raises the very exciting possibility to us that basically aneuploidy could be a driver of evolution. It basically allows the cell to roll the dice more often. One particular form of genome instability we were particularly excited about, and I want to highlight here, and that is this realization that virtually every single disomic yeast strain that we looked at had an increased number of RAD52 foci. What is RAD52? RAD52 is a protein that is important for DNA repair. Specifically, it's important for homologous recombination. And when you start seeing foci in the cell of that particular protein, that tells you that there's ongoing DNA repair. And so this increase in RAD52 foci was not again specific to the disomic yeast strains that were originally observed. We also saw that, again, these more complex aneuploid cells that we generate through inducing meiosis of triploid yeast cells, we also saw that there was an increase of cells with RAD52 foci. So a critical question that sort of comes from these particular studies is sort of, are there more breaks, or is the repair defective? We're working on this. Our data right now suggests that there's actually more breaks being made. We're trying to understand why they are being made. And so while we are still going into the details of trying to understand the molecular mechanisms underlying this genome instability, I want to highlight another very exciting finding to us. This was work done by my colleague Osami Niwa in Japan, and he for a long time has been studying the effects of aneuploidy in fission yeast. And his data too suggest that aneuploid fission yeast cells shown here on the right versus euploid cells shown here on the left, they too have a dramatic increase in number of RAD52 foci, raising the exciting possibility that these genome instability inducing events of aneuploidy could be conserved at least across yeast species. So obviously a very important question that we haven't addressed yet, but that are critically important, are these genome instability inducing events due to single gene imbalances that either interfere with DNA metabolism or chromosome segregation? There are examples for this in the literature. We know that when you change the stoichiometries of centrosome components, that can lead to chromosome missegregation effect. Or is this yet another facet of the general effects of aneuploidy causing cellular stress? There are examples for that in the literature also. We know that heat shock causes sensitivity to DNA damaging agent. So the general proteotoxic stress that we see in the aneuploid cells could also lead to increased genomic instability. So clearly, one way of thinking about how aneuploidy could impact tumorigenesis, as I said, by inducing genome instability, it may allow the cells to roll the dice more often. And so we are now basically interested in trying to understand if aneuploid cells basically are more quickly to adapt to various stresses that they're imposed on. So to address this question of the importance of rolling the dice more frequently. So irrespective of whether and how aneuploidy contributes to tumorigenesis, the gene-specific effects, genome instability-inducing effects, and many other possibilities. The fact remains the vast majority of human cancers are aneuploid. And so with that statement, there are two questions that immediately arise from this. The first one is, these aneuploid cancer cells proliferate really well. So they must have learned ways in which they can tolerate the adverse effects of aneuploidy in order to take advantage of potential beneficial effects. So the question is, they must have some mutations that allow them to tolerate aneuploidy. And if we understand what kind of mutations allow you to tolerate aneuploidy, that might actually help us to understand how tumors arise and how tumors evolve. So one question that we're particularly interested in is it actually possible to identify suppressors of the adverse effects of aneuploidy? And I'm going to show you a brief example in budding yeast that suggests that that is actually the case. And a flip side to this question is, can we identify enhancers of the aneuploid state? If we find drugs or genetic manipulations that specifically target either gene-specific effects in aneuploidy or the general effects of aneuploidy, well, maybe then that would be a way for us to develop new cancer therapeutics. And after we'll talk about the suppressors, I'm going to tell you about a study in mammalian cells that we've taken as a proof of principle that suggests that indeed such compounds do exist. So basically, we were interested in understanding the question, does aneuploidy, can we identify suppressors that allow aneuploid cells to grow better? The way we did this is very simply, we took some aneuploid yeast strains, they grow really badly, and we keep growing them until they grow better. And then we simply ask, what has changed, okay? So this is a growth doubling times that's a graph that shows how quickly various strains divide. And in red, you see the parental aneuploid strain. And in blue right next to it here, for example, those are basically evolved isolates that clearly now grow a lot better than the parental strain. So what we then do is we take these strains, we submit them to whole genome sequencing, and then identify the mutations. And then we generated a large list of mutations that allowed various different aneuploid strains to grow better. What was interesting is that among all these evolved isolates, we actually found 3 mutations multiple times in various different aneuploids. For example, we found a truncation mutation in UBP6 in both strains that had an extra copy of chromosome 5 or an extra copy of chromosome 9. We found mutations in VPS64 in 2 strains, and we also found a segmental duplication in multiple ones of them. So we decided to pick one of these and look at it in more detail. And so we started with UBP6 mainly because it was the first one we found. And so wonderful work by Dan Finley at Harvard Medical School had previously characterized this protein. They found that it is a deubiquitinating enzyme that's associated with the proteasome. The way you think about UBP6 is it competes with the proteasome. So it antagonizes proteasome function. And so the first question that we wanted to ask is, does deleting UBP6 actually indeed help these disomic strains, right? Is mutation of UBP6 actually responsible for the phenotype we observe? And so I'm showing you these experiments here. What we're doing is we're doing what's called competition experiments here. So we take the parental strain and we mark it in green here. And then we take the same disomic strain that's deleted now for UBP6, we co-inoculate them here at 0, and then we grow them over time, and then ask which part, what fraction of the co-culture is due to one strain versus the other strain. And you see very clearly that disome 5 and disome 9, deleting UBP6 really clearly helps them, which was good news because that's how we actually identified mutations. We furthermore noticed that there were 2 other strains that were also helped by deleting UBP6, and actually only one of the disomic strains actually grew more poorly. So indeed, deleting UBP6 helps 4 disomic yeast strains to grow better. And so Eduardo Torres, who did this work in the lab, developed the hypothesis that deletion of UBP6 increases the degradation of an unknown number of proteins and reverts the protein composition more back to that of wild type. Okay, sort of because deleting UBP6 antagonizes the proteasome, so now the proteasome is more active, it degrades excess proteins. So using SciLac mass spectrometry experiments, we can actually test that hypothesis. And again, this was work in collaboration with Steve Gigi. And what you see here is if you take 2 wild-type strains and you compare them, you get a certain distribution with a certain standard deviation. If you now compare a disomic 5 strain with a wild-type strain, obviously there's some extra proteins here, so the distribution gets wider, and your standard deviation increases. Okay? And what's really obvious, just from this bird's eye view, 10 miles up in the air, you can see if we now take a disom 5 strain that's deleted for UBP6, okay, and ask what's the standard deviation of that, is you see that compared to UBP6, wild type, disom 5 alone, it becomes more narrow. It doesn't reach wild type, but clearly things happen. So Eduardo was very curious to figure out, well, what exactly does happen to these cells? So he did a simple experiment. He basically rank ordered the proteins according to whether they're overrepresented in the disom 5 strains versus that are underrepresented in disom 5 strains. And then he created 3 bins. He created a bin where nothing changes, like within 1 standard deviation of the mean. Obviously, the vast majority of proteins don't change. Then there's a small group that gets upregulated in disom 5 versus wild type. And then there's a small group that gets downregulated. And he specifically asked, when I now look at these 3 groups, what happens when I delete UBP6 to these 3 groups? Okay, and that's shown here. That middle graph, where nothing happens, that basically shows you that deleting UBP6 doesn't really do anything to the proteins that didn't change in disom 5 strains in the first place. Okay, that makes sense. But what was very striking is, so these are the proteins that are overrepresented in disom 5 strains. When we now delete UBP6, you actually see that they get dramatically attenuated. Okay, so they're getting downregulated. And he could show that this attenuation is both due to transcriptional and post-transcriptional mechanisms. What was very exciting, and this kind of makes sense, right? So the proteasome works better, so all these excess proteins get degraded. What was unanticipated and exciting to us, that actually the downregulated proteins got attenuated too. And he could show that this was due to indirect transcriptional effects. Okay? So this basically led to a very simple hypothesis, 2 hypotheses. One possibility is that UBP6 attenuates the proteome of the cells that get improved by deleting UBP6, whose growth properties increase. The other possibility is that deleting UBP6 attenuates the proteome of all the disoms, but only the small number that benefits are the ones where proteins that interfere with proliferation are sort of cleared. And the guys that don't benefit, well, the reason why they proliferate poorly, deleting UBP6 can't really help them. So there's a very simple experiment that we can do to distinguish between these hypotheses. We can take a disome where deleting UBP6 doesn't make them grow better. Okay, so we delete UBP, and then the prediction is if hypothesis A is right, then nothing should happen to the proteasome in these guys. If hypothesis B is right, they should still get attenuated, but they are not being helped. So we take a disom 13 strain, we do exactly what we did with disom 5, and what we found was that, like disom 5, the proteins that were overrepresented in disom 13 were also attenuated by deleting UBP6. Interestingly, and I don't have time to get into this, the attenuation of the downregulated proteins does not occur. And we actually suspect that that's the reason why these cells are not helped by deleting UBP6. But what this experiment tells you is that deletion of UBP6 attenuates the proteomes of all strains. Okay? But disomes in which UBP6 clients interfere with proliferation are rescued by deleting UBP6. But disomes by which other proteins interfere with proliferation that are not touched by UBP6, obviously deleting UBP6 won't help them. Okay? So there's two general conclusions that I want to make here. Again, our data suggests that the ubiquitin proteasome pathway is really important for the life of aneuploid strains. And sort of a more general conclusion, yes, it is possible to obtain mutations that allow aneuploid strains to grow better. And sort of obviously identifying such mutations in the context of cancer will be instrumental in trying to figure out how tumor cells evolve from benign to more and more aggressive disease. So in the last part of my talk, I want to address the question of whether the aneuploid states of cancer can be used in targeted therapy. And Yun-Chi Tang, a postdoctoral fellow in the lab, asked a very simple question: is it possible to identify drugs or compounds that preferentially impair the proliferation of aneuploid cells versus euploid cells? And she initially did a very small targeted screen that again showed that indeed such compounds exist. She identified an energy stress-inducing compound called ICAR and a proteotoxic stress-inducing compound 17-AAG as having these properties. So initially, when we did this screen, we sort of, rather than screening 60 or 100,000 compounds at the same time, Yun-Chi sat down and asked a very simple question: well, what have we learned about aneuploidy over the years? We've learned that these cells are under proteotoxic stress. We've learned that they're under energy stress. And so Yun-Chi reasoned that if she now took drugs and compounds that also induce energy stress or that also induce proteotoxic stress, that could synergize with the aneuploid state and could create what yeast geneticists call synthetic lethality. Either condition on their own won't kill the cell, but if you combine them, you create what we call a synthetic lethal situation. So she used a small number of compounds, around 30, and started screening them. And I'm only showing you one set of data here, where she actually looked at this compound ICAR and its effects on cells. So the cells that she's using here are trisomy 1 cells, and she uses trisomy 13 cells. And then she basically induces cultures these cells under various different concentrations of ICAR. And I want to just focus on basically the open squares and the filled squares. So the filled squares here are wild-type cells treated with high concentrations of this energy stress-inducing compound ICAR. They still can grow, albeit very poorly. But look what happens to the trisomy 21 cells. Not only do they stop growing in the presence of these concentrations of the drug, they actually start dying. The numbers go down. Okay? And the same is true for trisomy 13. And then to a lesser extent for trisomy 16 and 19, these trisomies are trisomies of smaller chromosomes, so obviously the energy stress is predicted to be less. This data here basically shows you the same data as this graph, except now we normalize them to the untreated control. I told you in the second part of my talk, these trisomic cells have proliferation defects. They proliferate poorly. So to specifically look at the effects of the drug, what she did is she normalized the growth to 1 for both untreated, and then asked what is the percentage of the various growth with respect to 0. And you see very clearly, dramatically different effects for both trisomy 1 and trisomy 13. I'm not going to tell you all the details. We characterize these effects in more detail. We know that it's due to a p53-dependent stress response that gets induced in these cells. And we don't exactly know yet the mechanism by which trisomy induces this p53 response, but we're currently working on it. What was more important to figure out initially is to ask, well, these drugs that we found to be selective for aneuploid cells, do they also work for cancer cells? Okay? And so to address this question, what she decided to do is she decided to take two colon cancer cell lines, groups. Colon cancers come in two flavors. They come in a min flavor and they come in a syn flavor. The min flavor has low-grade, low-complexity aneuploidies, and the syn flavor has high-grade, high-complexity aneuploidies. So if our drugs indeed have some selectivity for aneuploidy, the clear prediction is that they should be more effective in inhibiting proliferation of syn cells compared to min cells. So that was a very simple prediction. And so she tested this prediction, and the data shown here, ICAR and 17-AG alone have not that great effects in the cancer cell line, so she combined the treatment. And in black, you see euploid controls. Then in blue and in green, you see the min cell lines, and then in orange, red, and purple, you see the syn lines. And you can very clearly see a differential effect between the min lines and the syn lines, okay? And so down here is the chromosome number and also whether or not p53 is functional or not. It was very interesting to see that actually p53 status did not appear to influence the sensitivity. So she subsequently asked whether she also sees effects in xenografts. How these experiments work is you take an immunocompromised nude mouse, you inject these cancer cells into both flanks, the min line flank in one flank, the syn line in the other flank, and then you treat the mouse with either control PBS or with the drugs combined, and you see both in the untreated, both sides, the min and the syn cancers grow equally well. But what was very clear is that in the presence of the drug, the proliferation of the syn cell line was dramatically induced compared to the min cell line. I want to be very clear here. I do not mean to imply that the only reason for this differential response is the degree of aneuploidy. I'm sure there's lots of other reasons why min versus syn lines could respond differently, but our data do indicate or are consistent with the idea that aneuploidy, the degree of aneuploidy, contributes to this differential effect. And so with that, I would like to conclude that aneuploidy could be a potential target in tumor therapy. So with this, I want to sort of end this part on cancer, and I want to summarize and sort of also discuss a little bit of what kind of implications our studies have on how we think about the impact of aneuploidy in cancer. Our data very clearly indicate that aneuploidy causes a proliferative disadvantage. It's bad news to have the wrong number of cells. And we propose that the proliferative disadvantage caused by aneuploidy needs to be overcome during transformation, during tumorigenesis, for the cancer to take advantage of potential beneficial effects of the condition. Our data also raised the interesting possibility that cancer cells may be more heavily dependent on the mechanisms that help cells deal with the stresses associated with aneuploidy. For example, proteotoxicity is something that's very prevalent in these cells. We would also like to propose that the phenotypes that are shared by aneuploid cells may provide a new therapeutic target. I've shown you one example, ICAR and 17-AG. Now we're actually in the process of screening large-scale chemical libraries for new compounds, unanticipated compounds that perhaps show this differential effect. And then finally, I think one way to think about how aneuploidy could promote tumorigenesis is by increasing basically adaptive potential by allowing cells basically to roll the dice more often. So on that note, I would like to again thank you for your attention. I want to end by acknowledging the people who did this work. The Aneuploidy project was started by Eduardo Torres many years ago. The transcriptional response work I told you about was by a graduate student, Jason Shelter. Anna Oromendia did the proteotoxicity and poly-Q work that I showed you at the very end. The protein aggregation work was also by Stefano Santaguida, and the hunt for drugs that specifically inhibit proliferation of aneuploid mouse cells was the work by postdoctoral fellow Yun-Chi Tang. Last but not least, funding. Thank you, NIGMS and the Howard Hughes Medical Institute. And thank you very much for listening.